would like to welcome all of you and thank you for joining us. And also thank Marnie Winter, Dr. Marnie Winter, for joining us and doing the presentation here. I think it's going to be a very, very interesting one for everyone involved. So you keep posted for that one. I will share my screen with you now. In the meanwhile, I can explain to you how it's going to work. So during the agenda, so we will have the first presentation from Marnie Winter, and during the presentation, you're going to be able to ask questions through the Q&A option inside Zoom. So everyone, if you take a look at the toolbar, you can see Q&A. If you click it, you're going to be able to ask questions. And you're not only going to be able to ask your questions, but you're also going to be able to upvote each other's questions, which means we're going to be able to ask the questions from Marnie directly, which have the most attention and which most of you thought was interesting or important from the presentation itself. So this is especially, this webinar is especially made so that you are able to communicate and you're able to connect with each other, especially in the single cell field. And so because of this, I would like to encourage you to ask as many questions as you can and as you have. Do not be afraid to hold back on these ones. As I said, we organize these webinars because we believe that single cell isolation is going to play a key role in the research in biomedical industry as well as biology. And because of this, we would like to provide a platform where researchers can connect and learn about each other's fields a little bit more, and also to just give an opportunity to people to connect with other researchers in the field as well. Now, our presenter here, Dr. Marnie Winter, is a biomedical engineer working to develop technologies for pregnancy applications. And her presentation is going to be also in this topic. She's based at the Future Industries Institute, University of South Australia, and she has been using our valve control equipment for quite some time now. And they've been capable of achieving very good results with this. And this is what she's going to be presenting about. So without further ado, I would like to give the word to her and let her share her screen with us.
All right, so hopefully you can all see my screen.
Yes, we can.
Yep. Okay, cool. Thanks everyone for joining. It's a pleasure for me to talk to you all today a bit about my research. And thanks also to Alex and Blint at CellSorter for asking me to come and speak to you all. I'm going to share with you a little bit about my work on the enrichment and then isolation of circulating fetal cells. I'm going to focus a bit more on the isolation as it's more in the theme of the seminar, but feel free to ask me questions if the enrichment aspect interests you as well. So a bit of background, in pregnancy, a lot of the time people will find out that they're pregnant and then they get sent for a heap of different tests to work out if the baby and indeed the pregnancy are healthy. In particular, what they're looking for, or one of the things that they're looking for is any genetic abnormalities. And normally they would do this and they would screen for this using ultrasound and also biochemical screening. And that combined with maternal risk factors and also family history would be used to determine if the pregnancy is considered low, moderate, or high risk. When the pregnancy is considered high risk, generally it would be recommended that the mother receives either a chorionic villus sampling or an amniocentesis, which are both invasive procedures that remove some of the fetal cells that you can then test and analyze the genetics of those cells. Importantly, these approaches carry with them a very small yet real risk of miscarriage. So you really don't want to be doing these tests unless you absolutely have to. In the last, I guess, decade and a bit, a new approach or technology has been implemented and is very widespread now, and that is cell-free fetal DNA non-invasive prenatal testing, or NIPT. And this test relies on looking for fragments of the baby's DNA that enters the mother's bloodstream. And then you can pick up that DNA from just a simple blood test. And it's really, really effective to screen for trisomies 21, 13, 18, and also the sex chromosomal abnormalities. In Australia, it still costs roughly $500, so it is quite expensive, and it's not necessarily offered to everyone, but it is a particularly widespread test that has very much revolutionized prenatal testing. However, cell-free fetal DNA is diagnostically limited. Indeed, it's not actually a diagnostic test, but should only be used for screening. And it cannot provide the full range of information that you would get from an invasive procedure. So you would detect roughly 25% of all abnormalities that you would identify based on an invasive procedure. Sometimes you also don't get a result back, and this is particularly important when you've got pregnant people who are obese and the fetal fraction of DNA that you can obtain is quite low. For some abnormalities, it has a relatively low positive predictive value. So for trisomy 13, it's 21%. And for other abnormalities, it has an even worse positive predictive value with a very high false positive rate beyond those common aneuploidies. So for example, for microdeletions, it could be up to 90%. And for this reason, generally, it's not recommended that this test is used to look for things beyond those more common genetic aneuploidies and common conditions. However, it's still an incredibly useful test. There has been huge improvements in cell-free fetal DNA in the last few years, and there will continue to be many improvements as we move forward. But to really go from where we are now, which is a screening approach, to something that is more diagnostic, you would need quite high and ultra-deep sequencing, which would likely be very expensive, even in the near-distant future. If we imagine this Punnett square where we've got the invasive procedures or CVS or amniocentesis up in the right-hand corner, we consider those a diagnostic test that must be performed if an abnormality is suspected. And then we've got the cell-free fetal DNA. It sits down here. We don't get much genomic information from them, but they're non-invasive, so they are particularly useful tests. And again, these should only be considered a screening test with diagnostic confirmation occurring once a positive screen has been found. The, I guess, ideal or the holy grail would be to have something that gives you a high genomic insight and accuracy while also being non-invasive. And in the field, a lot of researchers have been particularly interested in circulating fetal cells as a way of providing this holy grail. So what are circulating fetal cells? Well, these are baby cells that are found in the mother's bloodstream from around 6 weeks into the pregnancy. There's several different types of fetal cells that can be found in the mother's bloodstream. I'm particularly interested in those that are derived from the placenta, known as trophoblastic cells. And these contain a whole intact fetal genome, which we could use for genetic analysis. And importantly, the trophoblasts are larger than the white blood cells that surround them, so we can potentially use this to enrich them. These cells are very, very rare, so there's roughly 1 to 5 of them per mil of the blood, although the exact numbers are not well defined. And the numbers we know increase in some pregnancy complications like preeclampsia and trisomy 21. So this is just a bit of a background into cell-based non-invasive prenatal testing. So the presence of fetal cells have been reported a long time ago in 1893, where fetal trophoblastic cells were found in the lungs of women who had died of eclampsia. And there was significant research aimed at developing cell-based NIPT testing in the 90s and early 2000s. However, the results were ultimately quite disappointing, with some questioning if they were even really there. The reasons why the results weren't very good at the time was the fact that these cells are so rare, they're very poorly defined, they're also very fragile, and often they're apoptotic. There's a lot of biological uncertainty with these cells, and also combined with the emergence of cell-free fetal DNA, it led to a near complete stop of the field. However, in the last few years, the last decade, a better understanding of cell-free fetal DNA NIPT limitations, combined with the technological advances in the field of rare cell enrichment, which has really been driven by the circulating tumour cell or the cancer field, led to a recent reappraisal of the concept of cell-based NIPT. And in fact, very recently, there's been a technology that has actually been used within the clinic to be able to get these cells out and then analyze them. But there's still a number of, I guess, things we need to consider, and whether or not something like their technology would become more widespread is questionable. So really, there's still a push to be able to enrich and then isolate these cells with an efficient technology that could be used in a standard lab. And that was sort of the goal of the research. So as I mentioned, circulating tumour cell enrichment is really the drive of the rare cell enrichment field. And there's been a lot of technologies that have been developed to actually get these cells out of blood. And these cells they're cancer cells that have entered the bloodstream, and they can also give important information in regards to the patient. So I was particularly interested in a technology known as inertial microfluidics, which has been very successfully applied to the circulating tumour cell situation. And this was developed by our collaborator, Majid Warkiani, who's currently at the University of Technology, Sydney. And it's a really efficient technology that separates cells based on their size. Importantly, it's quite gentle on the cells, and it's also very cost effective, as it doesn't require, in the enrichment stage, antibodies. It's also very, very high throughput, so you can analyze more than 20 ml of blood in less than 45 minutes, which when we're talking about such rare cells like the circulating fetal cells, we need to be analyzing a fair bit of blood to be able to get enough cells to analyze properly. So this is the schematic of the approach. We've got the inertial microfluidic device in the center there, and then we've got the syringe pump. So normally you would lyse the red blood cells in the blood sample and then inject what's left into the device. It then goes round and round and round the device until it gets to a bifurcation point or a fork in the road. And the smaller cells, because they've been pushed to one side and the larger cells have been pushed to the other side, what happens is we can actually then collect the larger cells, and then we can analyze them and do whatever genetic analysis or molecular analysis that we want to. I would like to just briefly mention there is a commercially available FDA approved product that is quite similar to this approach, and that is the ClearCell FX. So just to make you aware of that, but the technology that I have been using and that was developed here is slightly different. So back to circulating fetal cells. So we decided, because there are so much similarities between circulating tumour cells and circulating fetal cells, that we could probably take this approach that had been so successful in circulating tumour cells and apply it to the circulating fetal cells. So I spent time optimizing the inertial microfluidic device to enrich the circulating trophoblastic cells. So they're the cells that are coming from the placenta. And this was fairly successful. So these are just some examples of some circulating fetal cells that we found from like clinical samples. The green cells, I'm not sure if you can see my mouse, the green cells are circulating fetal cells that are stained with a specific cocktail of antibodies that should be expressed by the fetal cells. And the red white blood cell down the bottom is a contaminating cell that we still end up with our enrichment approach. The second image here is a bit of a fetal cell cluster, and we actually found this from the blood of a patient diagnosed with preeclampsia. But how do we actually confirm that these cells that look like fetal cells and are staining like fetal cells are actually fetal cells? Well, we decided, and pretty much the easiest way to do that is to do fluorescence in situ hybridization and look at the chromosomal number. In particular, we took a male pregnancy, so a male fetus, and we looked for the X and Y chromosomes. So the X chromosome is in green, sorry, and the Y chromosome is in orange. So you can see clearly in this image here, in the center, we've got an orange spot and a green spot, which indicates we've got an X and a Y chromosome. The other cells close to that have two green spots, so they have two X chromosomes, and they're maternal contaminating white blood cells. The image at the top there is actually from a pregnancy affected with trisomy 21, where we found 3 copies of the 21 chromosome, which matches the clinical phenotype and therefore is likely a fetal cell. So when we actually looked at the numbers of what we're able to achieve, we recovered roughly 80% of the trophoblastic cells that we spiked in. So it's very difficult or almost impossible to work out how much you can recover based on clinical samples. So we were using a spiking model, and we get back around 80% of the cells. We deplete, and this is quite important, we deplete 99.5% of the white blood cells, which sounds really, really good. But in reality, when you're talking about many mils of blood and many millions of white blood cells, this still means that you end up with potentially thousands of white blood cells in the presence of a few circulating fetal cells at the end of it. The number of fetal cells that we found, we got roughly 0.62 cells per mil from a normal blood sample. And in pregnancies affected by trisomy 21, we get roughly 0.85, so slightly higher. And these numbers are still fairly low in what we would have expected. So how do we go from there to actually something that's a bit more clinically significant? Well, the real significance and I guess challenge is to actually be able to obtain a pure fetal cell population. And unlike CTCs, where you can enrich the CTCs and then perform an analysis and you look for specific markers that are expressed by the cancer cells, you can't do that in this instance because you're interested in the fetal cells, and they may, depending on the whether there's a genetic abnormality or not, look very, very similar to the maternal cells. So we can't use that to differentiate them. So you have to isolate the single cells or have a very pure population of those cells. So the workflow that we kind of settled on was we would obtain a blood sample and we would lyse the red blood cells. We would then inject the sample into our inertial microfluidic device. We would, after collecting the sample, fix and stain the cells. And we would stain them with a specific cocktail of antibodies, which would stain our fetal cells green and our maternal cells red, or the white blood cells red. After doing microscopy, we then used the CellSorter Piezoelectric micromanipulator to go in and pick up the individual cells and then deposit them within a little PCR tube. And so it might be a little bit difficult to see, but that white spot there is actually a cell that we've deposited in the tube, and we can confirm that it's in there. And then we go and do the genetic analysis on either a single cell or a pooled population of fetal cells. So just a little bit more information about how we went from staining the cells, and the cells were in a cell suspension, to actually picking up the cells. We used this 3D printed dishes that CellSorter made. And the idea behind that is you put oil and you have a very thin layer of cells in either PBS or media in the bottom. And then the cells, once they've settled, don't move around too much. So when you're bringing the manipulator needle in and out, it doesn't lead to the cells completely floating away. And so that worked fairly well for us. I'll talk a little bit more about that later. But what we found was when we started going and doing sequencing, so we started using an Illumina VeriSeq with MySeq, and that was developed for pre-implantation genetic screening. We were relatively successfully able to get these single cell profiles. However, sometimes with the clinical samples, we found that the quality of the DNA that we were getting was not great, and also purity was an issue. So we went back and looked at the timing and different aspects of the isolation approach and enrichment approach, and then also at whether we needed to fix the cells or if we could do something else to preserve or help improve the quality of the DNA, but also likely it's due to the nature of the cells themselves. They don't always have the best DNA. Purity was more of an issue when talking about the manipulator. And so what happened if we imagine again, we've got the little dish with the little wells. When we have a fetal cell that is right next door to a maternal cell or very close to a maternal cell, what happens is when you extract the fetal cell, sometimes you accidentally suck up the maternal cell as well. What we were doing in that instance is we would then deposit the fetal cell and the maternal cell into a neighboring well, and then we would push the cell out and it would sort of disperse, and then we could go in and pick up the fetal cell before doing the genetic analysis. So it became a bit more time consuming because we were double handling, but at least we were able to slightly improve the purity. However, there were some disadvantages with doing that. So sometimes we lost the cell, and sometimes we would have to wait for the cell to sediment again, which added to the time of the whole process. So we thought about how can we improve upon I guess, this process? And we thought about, well, it'd be really good if our cells were not floating around, but were actually attached to something. And then we thought about how we would be able to do that and actually successfully pick up the cells. And what we decided to do, we got a glass slide and we coated it with a silicon like substance called PDMS. We then treated that with sulfuric acid, which created a nano roughness on it and increased the surface hydrophobicity. We were then able to spin our cells using a cytospin or a centrifuge onto the surface of the glass slide. And then we coated that glass slide with a thin layer of oil. So what that looked like in reality was we had our nano rough surface, which was hydrophobic, and then we had our cell on top, and then the thin layer of oil. And then we were able to bring in the micromanipulator, and the micromanipulator itself contains the lysis buffer. We then deposited a droplet over the top of the cell with the lysis buffer. And this is what it looked like in reality. So you can see the single cell there in blue. And then we've got around that, we've got a droplet. And then we can also see the halo of the micromanipulator there. And we would just need to wait a short amount of time for the cell to actually lyse. And then we could pull up the lysed material and recover the RNA or the DNA. And then we can do whatever analysis downstream of that we wanted. We decided to do a droplet digital PCR and look for the RNA content, but it can also be done with DNA as well. So this is a bit of a technical information if anyone is interested. So with the micro manipulator, we were able to achieve very reproducibly sized nanodroplets. And that was easily achieved by changing the voltage that we applied to the piezoelectric head. And so it's important, particularly when you're talking about sequencing, the volume that you actually have your cell in, the lysis volume, changes the way that you handle the downstream and can also change the cost of performing that analysis. So having it in as small a volume as possible can offer some real advantages, which is another reason we were interested in doing this. So what it looks like in reality is we have our cell that we identify that we want to go and pick up. In this case, it's got the red box around it. And then we add the lysis buffer, the cell lyses, we recover the lysed material, and then we can analyze it. So we just did a quick analysis, I guess, to work out how much we were actually recovering and whether we were losing most or a lot of the RNA or nucleic acid content. The gap DH is a good example. So you can see on the right most graph, that's the single cell. So we put the single cell into the tube and we lysed that directly. And then we measured how much RNA we recovered. The best recovery, other than that single cell, was that of the PDMS that had been treated with the sulfuric acid for 90 seconds. And that gave us quite a rough surface, and it was the most hydrophobic of the surfaces, which meant that the droplet didn't sit flat on the surface, but kind of sat as a ball. And that meant that the surface area, the contact area between the slide was low, and therefore we lost less of the nucleic acid material. So just to summarize, there's still a number of challenges in the cell-based NIPT, both with the approaches that I've spoken about, but in the field in general. So it's really important to consider both the enrichment from the peripheral blood and the ability to recover both the fetal cells and deplete the white blood cells efficiently. And then the concept of having to actually isolate the single fetal cells, it's important to consider time and resource intensive approaches that are likely not compatible with clinical use. So it'll be interesting to see how the field develops and how widespread it can become, because cell-free fetal DNA NIPT is relatively straightforward and fits in with sort of normal or standard diagnostics, whereas the approaches that are currently being used for fetal cells are less streamlined. And then also, we want to, I guess, to better develop the genomic tests that we're able to run, particularly at the single cell level. It's difficult. The quality that you get is not always great. And the ability to be able to amplify and also perform the bioinformatics is really important. And then also, I just wanted to end on a couple of, I guess, considerations of what the field needs to be thinking about. And that's what maternal population would the test actually be useful for? Or would you want to screen the whole population, or would you just want to use this more comprehensive test to target specific high-risk individuals? And then also, what genetic conditions would you actually want to screen for, and what technology should be implemented? And that's really comes down to the cost-benefit analysis. I would just like to acknowledge Professor Benjamin Thierry, who I worked with on a lot of this work, and also Professor Majid Warkiani from the University of Technology, Sydney, who was heavily involved in the development of the inertial microfluidics and was also involved in optimizing it for the circulating fetal cells. And I would also like to acknowledge Dr. Mazam Rezai, who did a lot of the micromanipulator work. That's all I have to say. Any questions?
Thank you very much for the great presentation. So we have a few questions, actually, yes, and I'm going to read them to you in order as they came in. So the first one is it more difficult to isolate fetal cells from mother's blood than CTC isolation?
Yes, so CTCs, well, generalization because it is different in different cancers and with different patients, but in general, CTCs are a lot less rare than circulating fetal cells. And also in general, the downstream assays that you want to use to get something or like useful information for CTCs are a lot less, I guess, they need a lot less stringent samples. So potentially you can analyze a pooled sample. Like for instance, I've done some work where we used the inertial microfluidic device. We isolated the CTCs and then we just ran directly droplet digital PCR to pick up the cancer markers. We could never do that with the idea of circulating fetal cell non-invasive prenatal testing. It just wouldn't work. So yes, definitely a lot more difficult.
All right, thank you. In the meanwhile, I would like to highlight to anyone that you can still ask questions inside the Q&A button. Under the Q&A button, you can just ask the questions and we're going to read them. You still have time to put them in. We have 4 more questions right now. The next one is, how intuitive is the CellSorter system to use?
Yeah, so that's a good question. So we kind of used a hybrid of the CellSorter system and then our own microscope software. So we were using it a lot more manually than I guess is intended with the CellSorter system. And then I guess that's because the approach that we were using was a bit different to what the system had been set up as. So it was a lot more difficult. But when we use the CellSorter system for other applications where we're using it for in a fully automated approach, it is fairly straightforward. It takes a little bit of time to, I guess, get used to the software, etc. But when it goes and picks up the cells and then deposits them, it's just very satisfying. But yeah, I hope that answers the question.
Thank you very much. So the next question is, do you have a standard operating protocol which could be used by different labs looking to achieve the same results, or does the system require significant validation before use?
So I guess, is that question referring to the CellSorter system, I guess, not
I think so.
initial enrichment approach? So yeah, I'm not necessarily sure that our standard operating procedure would be the best one for other labs to use. I think we're still very much working on it with the idea in mind that we want it to be as quick and easy as possible. And it still is for us fairly time consuming to go and scan the slide, find the cell that we're interested in, and then go and pick it up. We have, I guess, published some of the work, which should give enough detail to go and do it. But also, I think in terms of the CellSorter itself, I would say that Belinda and the team were really fantastic in helping us, I guess, adapt it to our specific requirements.
Thank you very much. So the next question is, have you tested the viability of the isolated cells before performing assays?
Yes. So in the application of circulating fetal cells, these cells are not viable anyway, and the blood collection tubes that we use actually fix the cells. So we never investigated viability in that way. We have used picked up just sort of cell lines and then cultured those, which have been viable. But for us, the end goal was not the viability of the cells, because we can't culture those cells anyway.
Mm-hmm. I understand. I guess this answers a little bit the other question that we have here is, does this process damage the cells during the process?
Yeah, I mean, for us, the circulating fetal cells are so fragile that, and whether or not we recover less of them than I guess others do with other procedures, might be due down to the whole process, damaging and then losing the cells. I don't think so. The manipulator aspect was very gentle. The biggest issue we had was sometimes the cells would run away, and that's very much why we were interested in lysing on the glass slide, rather than trying to recover the cells and then lyse them to try and prevent them running away or getting lost. But that was kind of a rare occurrence anyway. But I think for us, the cells were a lot more damaged actually in the staining process, where we had to, add the different antibodies and then wash the cells, and they underwent centrifugation, etc. We seemed to lose a lot more cells in that process than actually in the picking up of the cells.
Thank you very much. So the next one is, what are the challenges you anticipate before this technique can be used as a routine clinical tool?
Yeah, I mean, that's a really good question. So there's a lot of challenges. So for us, really still looking at all three of those aspects of the enrichment. Can we improve the enrichment so that it gives us better statistics? If we're only looking for, 10 cells in the presence of 100 cells, or 100 contaminating cells, the odds are much, much better for us, and the ability to be able to pick up these cells is a lot better. Then also the actual isolation approach, it was still fairly time consuming. I'm very interested to hear what Alex has to say following this presentation to talk about, I guess, the more automated, from what I understand, version of the CellSorter system, because it was still fairly time consuming. The commercially available product, so the circulating fetal cell commercially available, or the one that's been clinically implemented, is very automated, but from what I understand, still fairly time consuming. But I think really the technologies that we need to be able to implement this routinely within diagnostic labs around the world is an approach that's going to be not that expensive and also not that time consuming, which is not a simple task. So there's still a lot to be done. And even in the genetics, there's still a lot of work to be done there, I think, to ensure that we're getting good quality DNA that we can get nice results from.
Thank you very much. And we have two more questions, actually. And the next one is, what material was used for construction for the spiral microchannel?
Yeah, so that was a PDMS microfluidic device. So it's the same. I can't say what it is because it's a long, complicated chemical name, but it's a very, very common like silicone structure that we use to make microfluidic devices. It's fairly common. The device itself, the spiral device, was made from a micromilling process. So we get a mold. We then pour this sort of silicon material into the mold, and then it forms the shape. We then peel it off and we can bind it to either a glass slide or more of the PDMS material. So it's fairly standard, straightforward microfabrication.
Thank you very much. So, and the last question so far that we have, if nobody else puts in anything in the meanwhile, the CellSorter system is pretty reliable. Can we use it on different samples like serum plasma?
I mean, I think it depends what you're looking for. I'm not sure what you would want to pick up in serum or plasma, or if you were just using it as a tool to, I guess, deposit small volumes of liquid, then you could use it on serum or plasma. I don't know if it would be the best tool to do that with, because it's really designed for more of the cell base. There's probably other tools that just dispense small volumes of liquid, but it would really depend on, I guess, your applications. I know for us, we would never consider using whole blood and looking for the fetal cells, because the odds would just be so stacked against us because those cells are so rare. We would very much struggle to find the cells without that pre-enrichment step. But there's other pre-enrichments that can be used. So there's, magnetic beads coated with antibodies, etc., that can be used instead of the approach that we used.
All right, thank you very much. And if we have no other questions, then I think this was the Q&A round for your presentation. So thank you very much again for joining us.
Thanks,
and for the great presentation. And we will continue with the bit of news from our side. So, and a few announcements from CellSorter side. We have very interesting developments in the last couple of months at CellSorter, and we wanted to give you an update on these things as well. You are free to ask questions from these sections as well if you're interested. I'm just going to breeze through it a little bit. So what we have is, just a second. Okay, so as you know, CellSorter was originally founded in 2005 by Balint Szabó and Zsófia Szabó in Hungary, who created the CellSorter systems. And during the years of development, after that, we arrived at the compactest of device prototype in 2020. Now, there is something very interesting that happened this year is that CellSorter has been acquired by the Semilab group, which is very relevant for us because we are receiving much more resources for development. And we hope this is going to accelerate also what's going to be available to you commercially pretty soon. And there is one more thing that is available right now, and it's very exciting for us as well, is the new device that we have. For you, if you're listening, you can see it behind me. This is actually the newest device that we will commercialize. The official launch is going to be this year in December at the ESCB Cell Bio conference in Washington. But you're going to be able to place orders already in an early bird kind of action right now, sale right now, and just a few information about it. These are our main users right now. So in these couple of years, since 2005, these institutes have been purchasing our instruments, and they've been actively using them as well. You can see all the papers that they've published using our instruments, such as also Dr. Marnie Winter from the University of South Australia, and all of the others on our website. I will show you the links for that as well later. Now, this is how the newest device looks like. So this is the compact desktop device. This is our most developed system so far. As you can see, everything is inside the casing in this situation. So it takes up a much smaller footprint, and it can fit onto a desk without any problems, as you can see behind me as well. This is a workstation where you could put it in, and it would be much more practical. And it's also the most automated system that we have so far. It doesn't even, it's capable of fully automation, and it's equipped with the piezo head technology right now. So these are the main points that we have about this system. It's going to be available on our website from now on. If you're interested in seeing the device in action, or if you're interested in more details about the equipment, then feel free to reach out to me or on our website as well. Now, about the applications that we currently have for this instrument. What we like to highlight, and what we think is very important, especially, is the rare cell isolation in this case. So these are all the papers, these are most of the relevant papers that were published using our instruments. But what we like to highlight is the rare cell isolation, especially in relation to Dr. Winter's presentation, because of CTC isolation. So what we believe this instrument will be excellent for is this, because in this scenario, it matters that you be able to isolate as much of the target cells that you have as possible, and that you're not losing them, and you're not losing them to also damaging the cells, and you're going to be able to harvest as much information from them as it's available. And thank you very much for joining us. There's going to be a feedback form at the end of this presentation. When you're closing the call, please don't close the browser. You're going to get it also in an email afterwards the call. We would really appreciate it if you could fill it out. And thank you very much for joining us. If you have any questions regarding the new instrument or CellSorter, then this is your time to ask it. I will just leave a few minutes for that, and afterwards I will close the call. I've got a question regarding the feedback form. The feedback form is going to be coming to you in an email, and you're going to also be able to see it just right after the call is over in the browser. It's going to pop up for you. What is the main difference between our new instrument and the old ones? So the main difference is, well, of course, the casing. So in this case, it's everything is inside the box that you can see behind me, and not in a scenario that you would see right next to me here. It takes up less space on a desktop, and it's also fully automated. So in this case, every single step of the way is going to be automated, and it's going to it has a even more intuitive interface for the software, and it's just mostly applied to in cases where you need to make it as hands-off as possible. All right, I can see no other questions, so I would like to thank you everyone for joining us today, and thank you again, Dr. Winter, for joining us and doing this great presentation. I will share the recording with everyone present here. Have a nice day.